All right, so now we're going to start talking about Punnett squares. Specifically about Punnett squares, we're talking about the crossing of genetic material, basically what you will pass down to your offspring. So for this video, we're basically going to learn about the basics of Punnett square, and we're going to be testing it out by completing the cartoon baby activity. So there's a few key terms that you should have learned by now, but we need to make sure you understand. So for starters, genotype and phenotype. So you've probably seen this by now when we write out and draw Punnett squares, heredity, and things like that. We tend to give them these two-letter, uppercase, lowercase definitions. So something's two-little letter thing is called their genotype. That's the gene they have. They have big B, or big B, little b. But then the phenotype, the genotype is the gene, phenotype is the physical representation of that. So for this case, genotype is big B, which codes for brown hair. So the genotype is big B. The phenotype is brown hair. So that's important to understand. So I'll ask you for the genotype or the phenotype, you need to know which is which. So, and then we have terminology for the genotypes. Big B, homozygous dominant. Now remember, dominant versus recessive alleles. Essentially, remember, dominant is called dominant because it is expressed. Recessive is not expressed unless, of course, there's no dominant. That may sound confusing, but let me show you. So, big B is homozygous dominant. Okay, so homozygous means the same. It's a big B and a big B. It's the two of the same thing. So homozygous dominant. Capital letters indicated dominant gene is the gene that's going to be expressed if it's there. Big B, little b, heterozygous. So homozygous means same, hetero means different. So a big B and a little b, that is heterozygous. Would any of you say heterozygous to somebody who knows this, they'll say, oh, so they have a dominant and a recessive. And then there's little b. It's homozygous again, but this time it's homozygous recessive. Remember, the deal with dominants and recessives is the recessive trait is only expressed if there's no dominant one present. If there's any dominant gene present, then it'll be expressed. So in this example here, big B, brown hair. So brown hair is the dominant gene. So big B would code for brown hair. Big B, little b will still code for brown hair because even though it has a blonde, so little b is blonde, even though it has a blonde letter in its genotype, it will still, since there's a dominant brown hair gene there, the dominant one's going to be expressed. So brown hair. But now with homozygous recessive, little b, there's no dominant gene to get in the way, so this person will be blonde. The blonde gene that's in the little b will be expressed because there's no dominant one to overpower it. And then the way we kind of arrange these things is we call Mendelian Genetics. Because remember Mendel, that guy who bred pea plants and kind of first one to figure out that we do these crosses. You set up what's called a Punnett square here with the parent 1's genotype up here and parent 2 along this side. Let's look at some examples. So let's change it up. So let's say capital F means you get freckles. Lowercase f, no freckles. So now we'll say that parent 1, parent number 1, their genotype is going to be big B, not B, big F, little f. What's this called? Yes, heterozygous. And what does big F, little f code for? That's going to give me freckles. Because, as we said, since there's a dominant freckles gene here, even if there is a recessive no freckles one, it won't be expressed because there's a dominant gene in the way. And we'll say parent 2 is big F, little f. Again, so these are two what parents? These are two heterozygous parents. Both have freckles. But both these parents are something we call a carrier. Why would they be called a carrier? Because they technically carry the no freckle gene. They don't express the no freckle gene, but they do carry it. So now let's set up our Punnett square. So parent 1's genotype. Parent 1's genotype is big F, little f. Parent 2 is big F, little f. Now these are going to combine. So parent 1 is going to go. So now this is how we do the cross. Parent 1 is going to give a big F. So in this column it's contributing a big F, and in this column it's contributing a little f. Okay, now this parent's going this way. In this column they contribute a big F and a big F. In this column they contribute a little f and a little f. And we're done. So we would say that these are the possible, these two parents mate, these two parents combine their genetic information, these are all the possible outcomes they can have. So they can either have 25% chance homozygous dominant. So they have a 25% chance of producing a homozygous dominant child, a child that has big F, homozygous dominant. They have a 50% chance they have a 50% chance of producing a heterozygous child, so a big F, little f, just like they have one of each. And then a 25% chance of producing a homozygous recessive. That being said, so both these parents have freckles, but they're also both carriers, they're both heterozygous. So what percentage chance do these two freckled parents have of having a child that doesn't have freckles? Right, 25% chance. So even though both these parents have freckles, because they are actually heterozygous, not homozygous dominant, they have a chance to pass on a freckleless gene. So even though both parents have freckles, they still have a 25% chance of having a kid who does not have freckles. Now, it is important to remember that this is called Mendelian Genetics. This works for learning the concept of genetics. This is not the rule how it makes. For example, earlier we did this one here, and you had brown hair or blonde hair. Is that how the world works? Is there one type of brown hair and one type of blonde hair? Is there no other in-between option? No. So that's what we're gonna look at: non-Mendelian genetics, partial dominance, co-dominance, and a bunch of other things we unfortunately don't have time to get into right now. But the point is, just to remember, this is Mendelian genetics. This is the basics. It gets way more complicated than this. There's a lot of fun mixtures, things like blood types, fingerprints, and so much more. But for now, this is what we're gonna learn. So this brings us into our activity, the genetics of the cartoon baby. So in this genetics of the cartoon baby, if you read through the instructions, it's basically saying you need one person to play the mom, one person to play the dad, mother, father. You can use one coin, you can each take turns flipping, you can follow the instructions, that's fine. Since I don't have a partner in this video, my left hand is going to be maternal, my right hand is going to be paternal. So the idea is, you just kind of pretending that we've gone after the square, we're kind of trying to see what it turns out to be. So for starters, doing freckles, according to the thing, heads represents a dominant allele. So a capital F, dominant allele, having freckles, and tails represent a recessive allele, recessive little f, no freckles. So, first things first, gonna have the maternal one flip, that is heads. Heads is that, so mother will contribute a capital F. Now it's time for paternal. Ooh, that's tails. Here's a lowercase f. These combine into the genotype of the offspring. This is like basically one of those boxes. So you don't have to write the dominant one first. It could technically be written little f, big F, but a lot of people just leave it as putting the dominant one first to make it easy. So that means that the genotype of the child they're going to have is big F, little f. That's called heterozygous. And that means they're going to have freckles. So, because they have heterozygous, they carry the gene for it. And so you're going to kind of go through this for the entire thing. Make sure you follow the instructions. Down here gets a little confusing. With this one you're doing, you're flipping it 6 times. Flipping the coin 6 times, recording dominant versus recessive, then you combine the total number of dominants, and that tells you where you land on the skin tone chart. The other one that might be confusing is up here. I'll practice with hair color. Now this one, like we talked about, it's already getting more complicated. So with this one, with male and female contributions, so we're doing hair color. Hair color's got a lot of options. See, we've got not just black and blonde, we got black, brown, blonde, and red. So let's do the 2 male contributions. So for A, male is going to contribute a dominant. So male is going to do a capital A. And for the B, male is going to do a capital B. Now, let's do female. Female is going to contribute heads. Female is going to contribute a capital A. And female is going to contribute, oh, wrong hand. There we go. Female is going to contribute a lowercase b. Meaning that So the genotype, they are homozygous dominant for the A and heterozygous for the B. So there would be capital A, capital B, lowercase b. Now you gotta kind of find out where that is over here. Now, you might have already noticed we have two capital letters. That means right here, this person, black hair. So we've got black hair. Now why doesn't that match any of the others? So you notice here black hair has A blank, B blank. And that's because, who can guess? Yes, because it doesn't matter what the other one is. Since there's already a dominant one there, it's gonna be that. So if our full readout is going to be A, B, B. So see these over here? These require double recessives on either A or B to trigger. So if you have big A in anything, because big A is going to be dominant, but you have little b, that's you have the recessive for B, so that gets you brown. If you have little a, big B, anything else, that gets you blonde. And if you have little a, little b, full recessive across the board, you have red hair. Now you're going to complete this whole activity, put it all together, and create your cartoon baby. So it has all the traits. You've gone over through the pages, you've gone through, you've created them, now draw it out. And that's how you basically kind of recombine your genetics, and this is the genetics of the cartoon baby.